Okay, so in this tutorial, I'm going to be going over DNA microarrays and mainly focusing on data visualization of the data and then analysis with R. We're going to be using RStudio, which is the integrated development software, and I'm just going to jump straight into it. So first, to even get it set up, you can just open up Anaconda Navigator. Once you get Anaconda Navigator open, you can install it. You can install RStudio. Now R is it's a statistical programming language specialized in statistics. So unlike Python, Python is a general purpose programming languages. So if you wanted to do most of the analysis, then you have to import all the packages. RStudio just focuses straight on statistics. And it has some other packages that we can use that are specially made just for microarray analysis, which is why we're going to use it. It just makes it a little bit easier to work with. So here you can you can scroll down, you'll find RStudio. You just click install, and then once you get it installed, you can switch to the RStudio application. Once it gets switched, you can launch RStudio from the Anaconda Navigator. Click launch. And then once this gets loaded, we can get started. So here we have it. Okay, so we're going to just start off by getting everything set up. So first we're going to import the data. It's microarray data, and we're dealing with, let's just open up, this is session number, to the GEO NCBI database. And you can read about the data here and what the experiment consists of. Basically, they have 9 samples. They're working on dendritic cells, and they hit them with some cytokines. And some of them are treated with cytokines. Some of them aren't treated with cytokines. And we're going to be looking at that. We're going to be sampling, clustering the samples, and using PCA analysis, principal component analysis, trying to reduce the dimensionality. But we're going to go in this one step at a time. So first, get RStudio open, and then I have this for you so you can copy and paste the code. You don't really need to code anything yourself. So first, if you don't have this already, install this BioC Manager to get this installed, and you can install GEO query. This is what we're going to be using. So once you get it installed, just like in Python, you can install a package and then you can include it, like import NumPy or import Matplotlib or import BioPython. It's the same format here with RStudio, but this time we're just going to do a library and then the name of the library. So we're going to paste this here, click enter, and then everything's set up. Okay, so that's good. And then we can copy this is the URL where we're going to download the data from. Copy this. Oh wait, it's this whole thing right here. My bad. Copy, enter, and then we're going to give it the file name where it's going to be downloaded. Copy, paste, and then we have this right here. So I'm just going to go ahead and we'll get one at a time. So then we're going to download the file. So now it's downloading the data. It's going to be in a matrix. So once this is downloaded, you can go ahead and then use the GEO library, and it's going to parse the matrix and the data that we have. So then we can start pulling information from it. So first, let's use get GEO. Copy and paste. And then this will start running. Okay, so now after running GEO or get GEO function on our data, you can just type dendritic data. You can get the features and the samples. The features, 54,675 are actually the number of genes. Next, we can look at the data with a box plot right here. Oh, I need to fix this, but make sure everything's spelt correctly. Box plot, let that load. While that's loading, fix this real quick. So we have box plot, and then we can just click zoom, and then it's going to pop up box plot. See, I can't really do much with the data looking like this. We can fix this though by doing a log2 transformation. You can run this here, or you can go all the way one page down and then run this code right here. We're just going to do the same thing from above, but it's going to permanently change the data. It's going to transform it, log transform it, and then we would plot the box plot. Click zoom, and then it's going to change it, and I can see that we have the interquartile ranges are real similar, the medians are similar. And so now we can start working with the data and start doing cluster analysis and PCA and stuff like that. So first what we're going to do is we're going to first filter out any unformative genes, and we're going to do this with the gene filter library. And with this library we're going to use the variable filter function. So we're going to import the library, use the variable filter function. We're going to filter the genes, and it's going to be filtered based off of the interquartile range. But first, let's just take a look at the data. So we have, here's the amount of data we have, and then we can copy and paste this whole code right here. And then now we have the features have been halved. In half, we went from 54,000 to 27,000. So the same amount of samples here. So just getting a look at how much we have left. By default, it's going to halve the filter, but now we have more informative variables, more informative genes that we can look at within our samples. So now we're going to start doing some unsupervised learning. We're going to try to figure out what samples are more closely related using the Euclidean distance. So just like how in k-means clustering from the last tutorial, I wrote from scratch the k-means clustering algorithm in Python just to show y'all what it's like to implement the algorithm and use it. But in this case, packages are already out there that already implement the algorithms, and we can just use them and not have to implement it by scratch every single time, which is useful. So to do this, we first need to create a distance matrix. So what we're going to do is we're going to use the distance function in RStudio to create the distance matrix, and it's just going to calculate the distances for each sample. So copy and paste, enter, and we have the samples right here and the distances. And then we're going to use hierarchical clustering to cluster the data using the hierarchical method. And copy and paste here, so we have 9 samples, 9 objects, and they calculate the distance from all 9 objects. And then we can actually plot the clusters, which using hierarchical clustering, we can create a dendrogram, which is sort of like a phylogenetic tree. And so we have each node, we have these nodes right here, nodes. And on these nodes we have the samples. So this group of samples right here, so this sample right here is most closely related to this one, then to this one, and you can say the same for this sample is more closely related to this one than compared to this one. Okay, this whole sample right here, this point right here is more closely related to each other than this sample's over here. So you can think about this as one big cluster, and this is also one big cluster. Always think about it as this is one cluster, this one cluster, just depending on what you're going to consider what the clusters are. But we also want to be able to see these sample names, not really giving us a lot of information about what these samples are, right? This doesn't really mean GSM 1282 or 3277. Let's change the name of that real quick. And then to do that, we can just run this code right here. And now we can click zoom. And then here we have changed the names. Now we can figure out, okay, so each untreated sample has clustered together right here in this group. And then the treated samples have clustered together as well. And we have right here noticing that we have three different treatments. We have the untreated, we have the this type of the IL-17A cytokine, and then we have the FNG. So we have those as a treatment, and then we just have the IL-17A as a treatment. And we can also highlight the color. We can add some coloring, add some more kind of annotation of the dendrogram to help see what's going on. So we can actually import another library here in Python. So going down, we can import the library WGCNA, import this library, and then we can figure out what the conditions are. So notice how there's untreated, then there's just the IL-17A, and then there's IL-17A and the IFG, so there's three different treatments here. And then we can slowly start color, say maybe we want to color them based off of different colors. So first we can say that all of them are blue, and then change them to the ones that are untreated will be yellow. And then we can actually graph this. Graph this here. And then this is the graph that we come up with. So here at the bottom we have all the yellows indicating the treated, the untreated, and then the blue is the treated. And we can go a step further and also identify the third treatment. So now we can say that the IL-17A and the IFNG are going to be represented as orange. And to change that, we can use this function again, copy and paste, and we get our resulting color change here. So here we have two of the same clusters of the same type of treatment, the IFNG, and then the IL-17A in this cluster, and then we have the untreated clustering right here. Okay, next we can also start extracting data from each cluster, and we can also specify how many clusters we want. So for instance, let's just show this with the red line. So the horizontal line going through the dendrogram is going to show how many clusters there will be. How is that so? Well, the lines that it intersects, so we have one intersection here and one intersection here. So that's going to be two clusters. If we were to lower this, say to 320, so let's change this to 320. 320. Oh, I know what's going on here. Have that off. There we go. Okay. Change it to 320. Notice how we have we have this line cutting through three vertical lines. The red line is cutting through three vertical lines, so we have three clusters. So this is treated as one cluster, this is treated as a cluster, and this is treated as a cluster. You can go further down too, and go further down. Okay, but that's just giving you an idea. So we can actually set it equal to 2. So 360 is basically saying that we're going to have two clusters if you're looking at the dendrogram. But you can also say k equals 2, and it's going to do the same thing. It's going to say we're going to have 2 clusters. And we can also take out this cluster and create a table using cut tree. So it's going to cut out, and now we have each sample. So these 4 samples are in one cluster, and this right here, the 2s, are another cluster. So there's 2 different clusters, 1 and 2. And then we can also turn it into a table to make it a little bit easier to see which one belongs. So we have 2 samples here. So we have 2 different clusters, 1, 2, and then in the columns it's showing where that one belongs. So we have the 2 cases right here. They're in the same clusters, the 2 different drugs that are added, and then the one different, right here is the one different drug right here. IL-17A is in cluster 2, and then IL-17A and F and G are in the second cluster as well, and then untreated are in the first cluster. So these samples cluster in 2 different clusters. So this one and this one over here. Okay, so moving on, we can actually create a heat map. So to create a heat map, we're going to need to first filter the data to reduce computation. So to do that, we can just filter by the variance of the genes. Instead of creating a heat map that's including all 54,000 genes, we can filter it based on the top 100 genes. So copy, run, and then it gives us the standard deviation. We can look at the variance here, but if you want, you can skip ahead and we can just focus on just creating the heat map. So here, we can create the heat map right here. Here it is. Here's the heat map. Okay, so you've already seen this one right here, this dendrogram. This is representing the sample. So each column is a sample right here. You look at the bottom and also it identifies it. So like earlier, the untreated samples cluster together, the treated samples cluster together. Then right here on the left hand side is the dendrogram of the genes, the top 100 genes. And then here's the gene names, or the gene IDs. The IDs aren't really that useful. In this case, we might want to change them to the gene symbols to get an idea of what gene it really is. We can change that too. And then you can obviously that there's some pattern going on. The differences in the expression levels for this group of genes over here compared to when you treat this dendritic cells, you have a different pattern expression happening here. And then these genes, so you can see that there's obviously some pattern going on in the expression levels between the genes just by looking at the coloration, okay? The expression levels. Each entry is an expression level, okay? So the column are samples, the rows are genes, and within that is the expression level of that gene. Okay, but we can start changing the colors and how we view the genes. Okay, and we can also add some of the sample pieces here, just like how we did above. We can add some more coloring to help us visualize where the samples are clustering together. Remember the yellow were the untreated. And we have this right here. But we do have some difference in the dendrogram. But we can move on. Okay, so now we can change out the gene IDs and we can replace them with the gene symbols. I have the code I've given right here. Enter, wait for it to load, And then here's our result. And you can see here is our results right here. So now we have gene symbols instead of gene IDs. Now we get a better idea of what gene is there, not just these gene IDs that aren't really meaningful to us with this visualization here. Okay, so moving on, we can also add some various colors here. You can install this and run it, and it's just going to change the coloring of the actual run. Now notice how the actual results here are different. See how when I'm looking at, or I can show you this one too as well, the clustering is different here. That's because of the default parameter that's used for the clustering. But not much to worry about in this case. We're not going to be focusing on like different parameters you can pass. Just creating it, kind of getting a general idea of what's going on here. Like we're creating a heat map here, and on the top we have the samples being clustered together, and then on the bottom and on the left side we have the actual genes being clustered together, and we can flip them out, and we can actually change the different colors to make a slightly different graph, visual graph. Okay, but next we're just going to move on straight to principal component analysis. So the idea with principal component analysis is to just reduce the dimensionality of the data set. So we have multivariable data, we have 9 samples, and if we want to plot them on a graph, it'd be pretty simple to plot 2 samples. We could have 1 sample on the x-axis, 1 sample on the y, and maybe we can also probably do 3 dimensions. On the z-axis, it could be the 3rd sample, but we have 9 samples, so it'd be pretty hard to visualize and create a graph that's going to represent 9 dimensions. So what we can do is we can apply the principal component analysis method that's going to reduce the dimensionality by trying to taking in each variable and figuring out which variables have the most information, and how we can break up the data into principal components and visualize it based off of those principal components. So it's pretty easy to do principal component analysis with R. So first we just run the PR comp. Okay, now we can just copy and paste this right here and run it, and then we get our Scree Plot. So the Scree Plot, this represents one principal component, one principal component. So these are the principal components right here. Okay, and for each principal component, they are in descending order. So this has the highest variance, and then all the way down to the lowest variance. Notice how the first principal component has the most variance, and then going down to the second principal component has the second, and then third, and it goes down descending. Okay. Usually we're going to use the PC1 and PC2 plot, because these usually have the most amount. These are explaining most of the data. Remember, principal components, they're constructed to find and determine what variables are most important, essentially. Okay, so we're going to use PC1 and PC2. But we can get out of here. Moving on, let's plot the principal components. And we're going to be using the principal components as our axis to plot the data. We'll plot it here. So notice how we have some clustering going on. So on the principal component, axis right here, we have some clustering going on right here. And then we have some separate clustering, but notice how we do have some clustering going on the x-axis right here. Now this variability, what you notice here on this x-axis, is more important than what you notice on the second axis right here on the PC1 or PC2 axis. Okay, but let's look at the percentage. Let's see how much percent of each principal component is making up. Is explaining. So here we have the same type of Scree plot, but in this case it's showing the percentage of the principal components. So this is saying that the percent variation is explaining like there's more than 25% of the data is being explained by this first principal component, and then same with the second principal component, and it goes in descending order. Okay, and then from here we can continue on, and we can make a more informative graph by using this again. Just copy and paste this, and then we can start coloring where they occur. So in this case, I'm actually going to need to redo this because it reset and then run. In this case, I always get this happening. So here we have the plot here, and we can look at it right here. And it turns out that with this plot, we have the treated and untreated clustering. So here's the untreated right here. And then we have the treated samples that are clustering right here. And we can also figure out, we can also see this happening in a box plot. So with the box plot, you can see that the data is being clustering here too, right here. So we have untreated, and then we have the two treated samples clustering real closely. Now this is more spread out, which makes sense because we look at the graph here. We have the treated being clustering together, and then we have the untreated not clustered together as well on the x-axis, but more so on the PC2 axis, or you can call this the y-axis. So here it is, here the clustering, but we definitely see some clustering with the treated samples. And then you can also see this with the box plot. You got the treated samples clustering together here, or in the same range, you know, in the same, almost close, pretty close medians. And then you also have the untreated sample here. And then we can also figure out what genes are responsible for where our plots are, where our samples are plotted at on the graph, on the PCA graph. And then here we have the 10 genes, top 10 genes that are responsible for making the graphs appear the way, making the graph appear where it is. So maybe we might be able to focus more on this particular graph. And that is the end of this tutorial. And I also have some resources here that you can refer to. It's a lot of information, and I just went over like briefly just creating the graphs, copy and pasting the code that I created here. But if you want to know more in depth, you can just refer to these resources that I have here that goes over more in depth than I did.